In this lesson, we'll take a brief journey through some of the early history of life on Earth. And we've got some momentous events in the 4 billion years on planet Earth, but we're going to pay special attention to this concept of endosymbiosis and how it transformed life on planet Earth. Of course, the key transition from a lifeless rock to a rock with life was the emergence of cells, simple types of cells like bacteria today. When cells emerged, they were capable of reproducing and evolving, and they spread across the earth, giving rise to the tree of life that we're familiar with today. So we want to think about life as a planetary phenomenon. Life is something that planets can produce. We're going to use this diagram here to capture some 3 billion years of Earth's history. And down here in the bottom left, we have a proposed site for the origin of life. This is still hotly debated, but these would be undersea hydrothermal vents roughly 4 billion years ago, where chemistry produced a living cell. Now, the chemistry involved here would have to have produced a set of molecules that we're familiar with. We know cells are made of molecules like lipids. The cell membrane is made of a lipid bilayer. Sugars like glucose are energy molecules. And proteins are composed of building blocks called amino acids. And then, of course, we have DNA with the 4 different kinds of nucleic acids, the 4 bases. So somewhere on Earth, these kinds of molecules were cooked up from complicated chemistry, and they self-assembled into the first life forms. This will be the origin of life. So we can think of these cells, as NASA says, as self-sustaining chemical systems capable of Darwinian evolution. On the tree of life diagram, then, Luca would be the last universal common ancestor. May not have been the first living cell, but it was the one that gave rise to descendants alive today. But what came before Luca was chemistry. Now, Luca would have been most similar to what we call bacteria today. These are the smallest and simplest types of cells. And when bacteria emerged from their rocky nurseries, they would have been feeding on molecules in their environment. So they would have to have the capacity to import molecules and use those molecules as a source of energy and building materials. We've learned in previous lessons that cells have protein channels that allow them to import large molecules like glucose, and then they have evolved teams of proteins to metabolize glucose and harness that energy in molecules of ATP to power the chemistry of life. Now, early on then, these first life forms would have been evolving new capacities. And we've learned in the previous lessons about how a population of bacteria might evolve the ability to eat a new food molecule. And one key process in helping us understand that capacity is gene duplication. Gene duplications can create new evolutionary possibilities. So here we have a circular chromosome, and here we have gene X. Well, during DNA duplication, mistakes can happen, and the entire gene can be duplicated twice. So now we have the original gene that codes for some protein that is doing some important job, but now we have a duplicate. And this duplicate then is free to accumulate mutations without loss of the original recipe for the original protein. So it's a way for life to experiment with new recipes to make new proteins. And in our example here, the idea was the gene that coded for the protein that imported the green food got duplicated, accumulated some mutations, and now made a protein that was able to import the orange food molecules. This would be a great advantage in an environment where there's green food and orange food, and perhaps the green food molecules are disappearing due to population pressures. Now let's return to this diagram. Here we have Luca. Now initially, Luca and her descendants were just eating molecules in their environment. But over time, natural selection would favor new genes to make new proteins that would allow a bacteria to eat different kinds of food sources. So we're going to have a period of the evolution of diverse kinds of bacteria. And we should note that at this early stage in Earth's history, there was no oxygen in the atmosphere, and so none dissolved in the oceans as well, or very little. So this was an oxygen-free environment. These cells were evolving in an environment with very little oxygen. But this was about to change. This lineage of bacteria evolved a set of genes to make proteins that could harness the sun's energy to make their own food, and we call this photosynthesis. So photosynthesis first evolved in bacteria, like these cyanobacteria today. Now, photosynthesis is complex chemistry, but it can be simplified in a reaction as you see here. The whole point of photosynthesis is to make this molecule here. It's a sugar, it's glucose, and this can be considered to be food. So photosynthesis allowed cyanobacteria to make their own food from simple materials in their environment. What were those materials? Abundant molecules like carbon dioxide and water. So using 6 carbon dioxide molecules and 6 water molecules, photosynthesis can produce 1 glucose molecule with 6 oxygen molecules released as a waste product of this particular chemistry. Now, the energy to power this chemistry comes from light energy. So cyanobacteria could capture solar energy and use that energy to do this chemistry, to produce food from simple materials in its environment. And this was a pretty cool trick for life on Earth, because prior to photosynthesis, all bacteria were eating molecules. They were using chemical energy in their environment. But with the evolution of photosynthesis, life tapped into solar energy. There was a very plentiful and reliable source of energy just 93 million miles away, our sun. Constantly bathing the earth in this energy, living things evolve the ability to tap into that, to capture that energy to power the chemistry of making their own food. So photosynthesis changed our planet. Oxygen, a waste product of photosynthesis, began accumulating in the atmosphere. And this would have a profound effect on the future evolution of life on planet Earth, because high oxygen in the environment was a requirement for the evolution of animals. Animals have very active lifestyles. They need a lot of oxygen to complete the metabolism of glucose in their mitochondria. And were it not for the evolution of photosynthesis, animals would not have been possible on planet Earth. But let's return back to the period of bacteria here. Initially, oxygen was toxic to many bacteria. Many species went extinct. But through gene duplications and mutations, some species evolved the ability to use oxygen to make food metabolism more efficient. They could extract more energy from their food, resulting in more ATP production. And so we're highlighting this red one down here. We'll just call it an oxygen-using bacteria. It's really good at breaking down its food molecules in a more complete way, but that chemistry required oxygen. So they were very efficient at making ATP energy molecules. So we can describe this early history of life on Earth as the age of bacteria, and it lasted about 2 billion years. Bacteria were experimenting with biochemistry to live in different environments and to eat different food sources, and indeed make their own food for some of them, like cyanobacteria. And if any space aliens wandered by planet Earth during this period of our history, and they visited our oceans and saw these little cellular beings, they might have had the following thought: we'll give these Earthlings some more time.